We can start now.
In medical genetics from Sir Gangaram Hospital. She has 14 plus years of experience at diagnosis and management of genetic disorders. Considering genetic, there are a lot of genetic tests available. It is valuable to understand which test to use when. Handing over to Dr. Kotecha to discuss this further. Thank you.
Thank you, Sushma, and a very good afternoon to all of you. A special thank you to Dr. Rahul Reddy in Hyderabad. It was his idea that we have today's discussion. There are lots of disorders in the pediatric endocrinology segment where one could suspect an underlying genetic etiology. But the focus of today's webinar is not disease specific, but rather to provide an overview of all the available genetic tests, to discuss the salient differences between the different test technologies available, to understand their advantages and limitations. For a few exemplary situations, we will be talking about a diagnostic workflow, but the overall purpose is to aid you as a clinician to try and pick up the possible best test for a particular phenotype or clinical presentation. There are very few branches where utility of genetic testing for treatment is known, and thankfully pediatric endocrinology is one of them. The best example, of course, is neonatal diabetes mellitus, where potassium channel mutations would lead you to go on and prescribe a high-dose sulfonylurea as compared to insulin. The ISPAD guidelines do state that next-generation sequencing, which allows the simultaneous analysis of multiple genes at a lower cost, and the cost is decreasing day by day and year by year, has become a feasible alternative to traditional genetic testing. Not only do genetic tests help you decide treatment in some cases, you could also predict and probably monitor response. One of the commonest conditions I feel that a pediatric endocrinologist sees is short stature. And of course, growth hormone is prescribed, but how do you predict what will be the final height, which is very, very difficult since height is influenced by multiple factors. These are from two different studies. The first one belongs to India, where they had 3 groups of patients, 2 having mutations, one in GHRHR and GH1 deletions, and the other is a group of patients with no gene defect. After 1 year of growth hormone, the ones with a homozygous deletion in the GH1 gene had the least response to growth hormone. This is another publication from Korea, which actually considered growth hormone response in patients with Noonan syndrome. Noonan syndrome is a known indication to start growth hormone. Noonan syndrome is one of the disorders which could be caused by multiple genes, what we term as genetic heterogeneity. PTPN11 is the most commonest gene, which is responsible for around 11 to 14% of Noonan syndrome. So these are bars which are comparing patients with PTPN11 mutations, that is the blue bar, with patients who had no identified gene defect. At that time, they had checked only 5 genes. Patients with a mutation in RAF1 and SOS1. And as you can see, the patients with PTPN mutations, though they started out doing well, they started waning after treatment. And this is something which is a known feature where we say they go on to develop some kind of insensitivity towards the treatment later on. What are the types of genetic variations that you're trying to pick up or what one could expect in a patient? You could have what is called copy number variation. You could have a single nucleotide variation, and you could have epigenetic changes. Epigenetic changes are disorders where there is no underlying specific gene mutation, but rather the expression of a particular gene is altered. Again, pediatric endocrinology is one of those fascinating branches where you have lots of epigenetic disorders to deal with. Examples being Prader-Willi, Silver-Russell, Beckwith-Weidman, as well as transient neonatal diabetes because of methylation abnormalities in chromosome 6. So the next few slides will actually cater to how do you identify these different kinds of mutations or genetic variations that we call them nowadays. Copy number variations simply mean that you can have a change in the number of chromosome, it could, or a particular portion of a gene. So your copy number variation could either affect an entire chromosome, a part of a chromosome, or even a part of a coding portion of a gene. The number could either increase or could decrease, and these are cumulatively called as gene dosage defects. You have a wide variety of tests to pick up these copy number variations, and they all cater to different kind of depths in the technology. The simplest way to detect large copy number variation, which still stays true, is a karyotype in a suspected patient for Turner syndrome. Of course, you could also consider the same in Klinefelter, as well as when you want to try and assign gender in a case of disorders of sex development, you could do a karyotype. The karyotype detects changes only if they are big enough. So they have to be around 5 to 10 megabase per huge for the karyotype to pick it up. But we do know, especially in Turner syndrome, that you could be having cases who have a mosaic cell line, meaning the presence of 2 or more population of cells with different genotypes within the same patient. While karyotype does detect some extent of mosaicism, so it usually detects mosaicism only when it is present up to 10% within a particular sample. Your mosaicism, your degree of mosaicism might be much less, and you might require other technologies. And one such technology is called FISH, or Fluorescent in Situ Hybridization. Very simply, what we do is we denature or separate the 2 strands of DNA. We then tag one of the strands with the probe, which is fluorescently labeled, and then we check for the fluorescence under the microscope. So if I want to do, and this is the FISH result of a patient with a mosaic Turner syndrome, my FISH probe targets the centromeric region of chromosome X. And you can see here there is only one green signal, and here there are 2 green signals, meaning that the karyotype of this patient would be, or the chromosomal complement would be 46XX, 45X mosaic. So if a suspicion of Turner syndrome persists even after a normal karyotype, you should go ahead and do FISH. There are international cutoffs to the number of cells that you can count, and the reason why the difference exists is the stage at which we stop the mitosis. So a chromosomal karyotype is done in a metaphase stage, whereas the FISH is done in the interphase stage where your chromatin has not yet been condensed into chromosomes. So with FISH, it is said that ideally you need to count 200 cells, and if all 200 are normal, you could safely say that there is no mosaicism. But however, it is also important to know that if your suspicion still persists, it is probable that just counting 200 such cells in blood may not be enough. You might be dealing with a tissue-specific mosaicism, and you might require to pull samples from other sites. The commonest samples that we recommend are buccal swab and skin fibroblasts. Similarly, you also need to test for the presence of Y chromosome in a patient with Turner syndrome. For that, you could use the FISH for the Y chromosome. And it's very important, again, to test multiple tissues if a case of Turner syndrome is having a virilization and your FISH for Y on blood turns out to be negative. But the most important part is that FISH is probe dependent. Now, how does this affect the performance of your test? This is an article which described a patient where there were gene dosage defects, and it depicted the usefulness of an array technology for the same. This was an 18-year-old female with primary amenorrhea, delayed puberty, a hypoplastic uterus, and a hypogonadotropic hypogonadism with a 46XY karyotype. They went ahead and did the FISH for SRY, and both these red signals belong to the probe region of the SRY, and their presence states that the SRY was present, so the results were normal. They went ahead and did a microarray, and they did find a partial deletion in the SRY gene. So why were the results discrepant? It all depends on where your probe is targeted. So this is the SRY gene. The solid lines conform to the FISH probes. And as you can see, it starts from here, one probe, and this is the spacing for the other probe. Whereas the deletion of this patient actually lay here. So it was in the adjacent region. So it was in an atypical region compared to what we would commonly find. And that is why the results were normal on a FISH assay. This just states that when you have a clinical phenotype, a negative result, rather than looking out for other causes, it's probably more important to understand the intricacies of the test, and then you can just go down deeper and you might end up with the same results that you were expecting the first time. So what is an array? A microarray is something which helps you to look deep inside into chromosomes, again looking for copy number variations basically. You have got two kinds of array technology. One is an array comparative genomic hybridization, or array CGH, where basically you take a patient DNA, you take a reference DNA and compare both together and find out whether there are copy number variations in the patient. The other array technology, which is gradually becoming more popular because we have other uses of the array in genetics, is what is called a SNP array, or a single nucleotide probe array, where on a slide you have got probes for single nucleotide polymorphisms. Single nucleotide polymorphisms just mean that at a particular point, what does a person carry? Does he carry nucleotide A, nucleotide T, nucleotide C, and nucleotide G? So of course, if you go into genetics, we just know about the 4 nucleotides which are responsible to generate a genetic code and code for amino acids. So at a particular position, you could have adenine, you could have thymine, you could have cytosine, or you could have guanine. And this is what the SNP array looks at. When would array be indicated? If I go along the same path of disorder of sex development, it is said that when you have something which is not completely explained by the phenotype. So if you have a child with ambiguous genitalia, but if the child has a malformation in a system other than the urogenital system, or if the child has additional features like this child who had dysmorphic features, failure to thrive, and developmental delay, which cannot always be correlated to the defect, then it is possible that you're dealing with a complicated defect which not only involves a single gene, but probably involves multiple genes. This child underwent a microarray, and the results suggested that there was a deletion on chromosome 4 and a duplication on chromosome 10. This is a pictorial presentation of the same microarray. They are numerically arranged. So this is chromosome 4, and the red bar here indicates a deletion. on the short arm of chromosome 4, which is designated as 4p, and the blue line here indicates a duplication on the long arm of chromosome 10, which is called 10q. Now, when you're considering an array platform, there are multiple arrays which are available. The difference between these arrays is how deep they look into the chromosome. So what is the size of the variations that they pull out? So you have a low density array, you have a medium density array, you have a high density array, and the highest density is possibly what we call an exon array. How do these arrays differ? Or their densities basically differ on how many probes are there and where are these probes placed. This is a pictorial demonstration of a low density array. And here you can see that there is a probe here and there is a probe here. So yes, if there is a copy number variation here and if there is a copy number variation here, the array technology will pick it up. But if there is something in between this, it will be missed. The exon level array on the other end is a very high-end array where literally there are no gaps and we can pick up the smallest of the copy number variations. But as the saying goes in genetics, more is not always the better. So the deeper you go in, you might come across changes, but you're not very sure whether that change actually is disease-causing or not. We now do understand that there are a lot of minor changes which are present in normal population, and unless you accumulate large amounts of data, it is impossible to say whether a small deletion of probably 100 kilobase pair would be important or not. So what is the kind of array which is usually utilized in the postnatal or pediatric subspecialties? We recommend something in between these two, what is called as a medium density array. The low density array is basically targeted towards prenatal testing because we want to only pick up bigger changes and we want to avoid a variant of unknown significance. The medium density arrays, one of the commonest ones is the Affymetric Cytoscan 750K, will provide you results when you have a deletion which is around 200 kilobase pair and duplications which are around 400 kilobase pair, which is good enough in the postnatal period. Of course, even with such resolutions, you could come up with a variant of unknown significance and you might have to test further to find out and understand the significance of your findings. Now, copy number variations, what I have discussed till now were related to chromosomes. You could also have copy number variations within a gene. So a portion of a gene might be extra or duplicated, or a portion within a gene may be missing or deleted. One of the technologies which picks such copy number variations is called MLPA, or multiplex ligation probe dependent amplification. Another technology which picks up copy number variations is a PCR, but a simple PCR only picks up deletions. It does not pick up duplications. A quantitative PCR, on the other hand, picks up both deletions and duplications. MLPA is a comparatively common investigation, at least for genes where copy number variations are common. And one such common example would be congenital adrenal hyperplasia because of 21-hydroxylase deficiency. So if you have a patient with CAH because of 21-hydroxylase and you sequence the gene, the chances that you will get an answer is only 70 to 80% of cases. In the rest, where you do not find any genetic variation, you need to go ahead and test for copy number variations via the MLPA technology, and there is a high possibility you will detect one, like what has happened in this patient. This is actually a mother of an affected patient, and that is why the deletion is heterozygous. But you can see the normal dosage should be 1. It has decreased to 0.5 to 0.59. There's always a range to it. And this would suggest that the person in question at least has one deleted copy of this gene and hence could go on to have an affected child if her partner also has a problem in the same gene. This was a parental testing done in a family who had lost already 3 children to CAH. A different version of the same technology, which is called methylation-specific MLPA, is what is available for Prader-Willi syndrome. Now, in many cases, I have seen that in suspected Prader-Willi syndrome, the first test to be ordered is a FISH for deletion. While deletion is the commonest genetic mechanism to cause Prader-Willi, it is only responsible for 65 to 75% of cases. So if you go into Prader-Willi is one of the imprinted disorders where the gene is only expressed from the paternal chromosome and the gene is not expressed from the maternal chromosomes. There are three ways by which a child can have Prader-Willi syndrome. Either there is a deletion of the region which is carrying the gene on the paternal chromosome, which is by far the commonest, or the child, instead of inheriting one paternal and one maternal chromosome, inherited both maternal chromosome 15, which we call uniparental trisomy, or it is possible that there is some problem in the imprinting center on the paternal chromosome, and hence the gene which should have been expressed has actually been silenced. Now, what is the most cost-effective way of detecting all these mechanisms? Because any of them could lead to Prader-Willi. And how are you sure that if you test by one mechanism, you have excluded Prader-Willi or not? That is where MS-MLPA is very important. This is from one of the papers which originated from Australia, which is a beautiful paper discussing all the different molecular technologies available in Prader-Willi. And this was their suggestion for the most cost-effective way of testing. You just start with MS-MLPA. It looks at deletions. It is able to comment on a possible uniparental disomy as well as imprinting defects. So if it is negative, the chances that the child has Prader-Willi syndrome is very, very unlikely. If it is abnormal, you are very much likely to be dealing with a case of Prader-Willi syndrome. The further limb of the testing is only important when we are considering prenatal testing, because methylation patterns might change in the fetal tissue. So we go ahead and find out what is the exact mechanism and what are the chances that they will recur. So for the clinical practice for an endocrinologist, probably just doing an MS-MLPA would suffice in cases of Prader-Willi syndrome. Coming on to single nucleotide variations, the first cartoon is that of a gene structure. You start with a 5' end. You've got the promoter region. Then you have coding portions, which are called exons. And in between coding portions are non-coding portions, which are called introns. Once there is a stop codon, no further protein is made, and the rest of the region is called an untranslated region. Usually when protein synthesis occurs, the intervening introns are spliced out and only the exons join together to form a mature protein. So this is a wild type or a normal sequence with this arrangement of nucleotides in this manner, and you could have a change at just one point of the nucleotide, and that is why it is collectively called point mutations. Now again, point mutations could be of different kinds. It could be silent, meaning that though the letter changed, so from T it became C, the coding amino acid remained the same. It was arginine on that position, and it still remained arginine on this position, and that is possible because the amino acid code is degenerate. So you've got more 3-letter codons coding for, you've got multiple such codes coding for a single amino acid. Other type of variation is what is called a stop variation. So the change is so huge that no further protein is made. This usually is expected to produce enough problem in the protein structure to cause disease. But again, it is important where the stop variation is present. While usually we expect most of the stop variations to be disease causing, supposing you had a stop variation in your patient just adjacent to where the protein is getting terminated, I would not be very sure unless I do functional studies where the loss of just this much amount of protein would cause such a significant effect that it would lead to disease. And that is why it is not uncommon that in genetic reports, though you have a frame shift mutation or a termination mutation, and we do state that the protein has been truncated, we still call it as a variant of unknown significance. The other common change is a missense mutation, where again there is just a change in a single letter in the code, but this has led to change in the amino acid. So arginine has either become proline or arginine has become cysteine, and it is possible that that change might cause a problem in the protein. You have other defects like insertion and deletions, and these again could be in frame or out frame depending how many nucleotides are affected. So whenever nucleotides are affected in multiples of 3, because that is what forms a genetic code, 3 letters together, we call it an in-frame deletion. Means that though there has been a change, the change has been in such a way that it just picked in one more amino acid, and you're ultimately making some amount of protein. If, however, there is only an insertion of 2 nucleotides like that they have shown here, it will cause a drastic change in the genetic code and affect protein formation, and that could lead to more disastrous sequences. How do you look at all these disease variations? Collectively, they are called single nucleotide variations. You could go on to Sanger. Sanger is a gold standard. Even now, in many cases, we do confirm variations detected by NGS through Sanger. Why? Because Sanger is a very, very specific test. I have one gene. I know it consists of 10 exons. There is a particular portion of the gene which is very difficult to sequence because of its structure. But through Sanger, I can design multiple kinds of primers, and I can make it possible to read that difficult to portion region of a gene. The only problem with Sanger is it does one gene at a time. So it might not be very good for disorders which we know can be caused by more than one gene, or what we dub in our genetic language as genetic heterogeneity. The next is called a multi-gene panel, which usually means that you are pooling genes which cause a similar phenotype or genes which belong to a similar pathway. Like we can take up the example of adrenal steroidogenesis or neonatal hyperinsulinemia, and you study all of them together. A step beyond that is your clinical exome or Mendelium as it is called. What does this mean? I pick up my Bible for genetic medicine, that is the online Mendelian Inheritance of Man, which is a freely available curated website. And I pick up all the genes which are currently associated with human disease. The term current is very, very important. Today, I might have 5,000. Tomorrow, I might have 5,200. The counts keep on changing. And I just study those many genes to try and come to a diagnosis. A step above that is whole exome. That is, again, I go to OMIM, but I am not bothered whether a particular gene has actually been associated with human disease or not. I just pick up all the 19 or 20,000 genes which are present and sequence them together. And the last, of course, is whole genome, where I am looking at coding and non-coding portions of the genes. So what do I mean by this? Now, all these technologies, barring Sanger, are comprehensively called next generation sequencing, and multi-gene panel, clinical exome, and whole exome test for only coding portions, exons, and regions around the intron-exon boundaries. Why have such limitations been kept? Of course, the more the test technology evolves, the more number of genes, the more number of regions that a test reaches to, the costlier it becomes. There is a higher chance you might end up with a variant of unknown significance. But most importantly, 85% of mutations across different disease specialties are usually present in exons. And that is why most of the testing technology has concentrated on the coding portion. The genome, on the other hand, looks at introns, that is the non-coding portions, exons, intron-exon boundaries, untranslated regions, as well as the promoter regions. So yes, you delve very deep into your genome, but at the same time, it would be very difficult for me to prove that a variation which is lying 100 base pairs away from a coding portion is actually causing effect and is causing disease. So it requires a lot more post-test functional studies to prove that it is truly disease-causing. Why NGS? Very simply, you have phenotypes which have more than one gene, which do not always have very good clinical indicators. Of course, if you have a child with suspected congenital androgen insensitivity syndrome, you could boil down your test request to a single gene or two genes. But if you have a child with just a disorder of sex development and the initial hormonal testing is not getting you anywhere, you could be dealing with around 61 genes in a case of 46XX DSD, and you could be dealing with around 62 genes in a case of 46XY DSD. So when will you begin? When will the patient get an answer? And how do you know that you have sufficiently covered all the genes, or till when has the patient to keep on testing? And that is where if you simultaneously analyze all these genes together, there is a higher chance that you will hit upon an answer in a cost-effective manner. Now, whenever you consider NGS, which kind of NGS you should be considering? So these are the questions that you need to answer before considering testing. The first and foremost you need to answer before embarking on any kind of genetic test: is there a genetic basis for the disorder concerned? Is it probable that the child with short stature in front of you just has a familial short stature? We know that there is an underlying genetic basis even for familial short stature. The chances that you will hit upon it with routine available testing, including whole genome sequencing, is very, very low. How well is its genetic basis defined? Are all the genes known to cause a disorder being defined? Can I just say that if I test 9 genes, I have finished with the workup? If it's negative, I have excluded the disease or not? And is there a difference in how the genes which are implicated in a particular disorder covered between the various tests available? What do I mean by this? So again, you have targeted panel sequencing, you have whole exome and whole genome. Ideally, I would say that pediatric endocrinology is a branch where you could actually offer targeted panel sequencing to a lot number of patients. You take up neonatal diabetes, you take up adrenal disorders, you take up glycogen storage disorders. All of them could be sequenced through a targeted sequencing panel. But what is an ideal targeted sequencing panel? An ideal targeted sequencing panel goes along the lines of Sanger. So I first sit, review literature, find out what are the genes which are associated with a particular disorder, probe into their structure, see whether there are particular areas which are difficult to sequence, go ahead and fashion my chip in such a way that I cover all the genes completely. Unfortunately, today I made such a panel for disorders of sex development. I included 20 genes. 6 months down the line, literature now says that there are 10 more new genes. I will have to fashion the entire chip again. So it's not at all a cost-effective procedure for the laboratory. And so, of course, it does not turn out to be cost-effective for the patient. So what happens behind the scenes in most laboratories? This is what happens. We do a broad-based test. The most common test, which is done by most labs, is a clinical exome mendario, where they check anywhere between 4,000, 5,000, 8,000, depending upon the chip that is fashioned. And then we just take out those genes which are involved in a particular phenotype. So if I take glycogen storage disorder, I pull out only those genes which are known to be associated with hepatic glycogen storage disorder. I analyze them for a possible genetic defect and give out my report. I would be doing the same even for osteogenesis imperfecta, osteopetrosis. Now, the biggest problem is when I do a clinical exome, I'm covering more genes. And as you can see here in the bottom, coverage. Coverage means what? It means that supposing a gene has 10 parts, are all 10 parts completely covered? And I'm only talking about coding portions. So that is what I promised you in an exome. Unfortunately, how much ever data I generate, supposing I take up 11 patients, all with osteogenesis imperfecta, I put them through the same process. I have not changed my process at any stage. It is possible that the same gene panel generated through the same route might give me 100% coverage for all genes in 9 patients, And in the next 2 patients, there might be some genes which are undercovered. Once I put my sample into a machine, there is no saying how much data will be generated. I have some control to it, but there is no saying where the data will go and hit. There is no way that I can target the data to a specific area because I have not designed my test to be so specific. So that is where the difference lies. Now, again, it's important to understand that you are going to find these discrepancies across different genes, across different patients. But if you have clinically phenotyped and boiled down to probably 1 or 2 genes, you could cross-talk with the laboratory after getting the report saying that, see, this portion is uncovered. It's very important for me in disease, and we will go and fill it up. However, if your phenotype is very broad, like a disorder of sex development, and I say that out of 61 genes, 54 of them have been covered up to 95%. 95% is the cutoff, which is accepted across most international laboratories, and a few of them have not been covered even up to 95%. It's not practically and economically feasible for me to cover all the regions. So there are going to be data discrepancies, and as technology gets better, probably the data discrepancy will go off because we might be able to generate more and more data at lesser cost. The more amount of data you generate, less the chance that you're going to have problems in your coverage, but the more amount of data I generate, I directly increase the cost of testing. It's also important to be well versed with the content across panels. If I take up, and this is an example, I have taken up this data from 3 well-reputed labs in the West. All of them have called their respective panels as panels for neonatal hyperinsulinemia. Lab 1 tests 9 genes. Lab 2 tests 18 genes, and Lab 3 is the external lab, which has a sequential testing, where in the first case, they do ABCC8 and KCNJ11, and if that's negative, they do a 20-gene next-generation sequencing. Why is there so much discrepancy when all labs pick up their gene lists from Omen? For the simple reason that what is the thought process behind designing a panel? And of course, what does a clinician want? Do you only want to look at genes which have been robustly associated with the disease phenotype? And when I mean robust association, in genetic terms, it has been said that a gene has been implicated in a particular disease and has been seen at least in 3 different similarly affected families. When I replicate it in 3 different similarly affected families, I would say that such a gene has a moderate gene disease association. So yes, all labs will include those genes which have a strong gene disease association, but some labs go a step further and include every gene, even if it has been reported in a single patient with such a phenotype. Now, of course, this is all done basically on the clinician's needs and their phenotypes. But when a gene has been implicated, so supposing this is just an example, it's not true. I pick up PMM2, which classically calls CDG, and I say that in one patient, it caused isolated neonatal hyperinsulinemia. According to the American College of Medical Genetics, I cannot tell you that it is a gene so common that I should be looking at this gene in every patient with neonatal hyperinsulinemia. The same goes, the more you increase, then the content of your panel increases, though the chances of getting an answer do increase, there is also an increased chance of getting a variant of unknown significance. If you include rare genes into your panel, there are many genes which are called genes of unknown significance because they have only been reported in one patient or one family, and any variant that I detect in a gene of unknown significance will be called as a variant of unknown significance. So the biggest problem that I consider with panel testing is that there is so much discrepancy across panels that it is impossible to say what is good and which one you should pick up. And most importantly, and you have to be well versed with all the updates in genetics to pick up the robust panel for your patient, which of course is even a task for a geneticist like me. Are, are ethnically important genes covered? This is another question you should be asking, because if you simply take the example of Modi, GCK, HNF1 alpha, HNF4 alpha, and HNF1 beta are the main genes implicated in West. In the Western region, they say that ABCC8 accounts for less than 1% of cases. But in a study from India done by Dr. Mohan, they showed that HNF1 alpha and ABCC8 are in fact more important in the Indian subcontinent, and GCK is not so much. So again, all these small points will make a difference when you are considering a panel testing. It's also important to understand that a diagnostic yield depends upon the current understanding of genetic basis of a disorder or a clinical presentation. One of the best examples in endocrinology would be short stature, and obesity would be another. Both are disorders which could either be inherited in a monogenic form, at least the severe or extreme types, and of course they also are well known to be inherited by multifactorial inheritance, where we say there is some amount of genetic susceptibility as well as environmental factors which are playing a role. In fact, more than 600 genetic variants have been detected which are involved in human height. But this again only explains a very small fraction of the phenotypic difference. You could be also dealing with a condition like familial short stature because of agrican mutations or acan mutation. It's an interesting gene. And it's very important because when you consider couples, there's a very high chance that a person with a short height will go on to marry a spouse with, again, a short height. Now, if you have two proportionately short stature individuals, because of agrican mutations or acan mutations, these individuals could go on to have a child with a skeletal dysplasia. So how does this happen? When one of your copies, so all of us have two copies of the acan gene, when one copy is altered, it only causes proportionate short stature or familial short stature. But if a child inherits both defective copies from the parents, the effect is much more and the child ends up having a skeletal dysplasia. You also could be dealing with what we call as oligogenic inheritance, that is different defects across multiple genes in a single pathway could culminate to cause either obesity or short stature. So broadly to say, both short stature and obesity are very difficult phenotypes to handle, and there is a very high chance that whatever depth of testing you do, we might not be able to come up with a cause unless it's a syndromic obesity, unless it's a severe short stature like microcephalic primordial dwarfism, or there is an underlying skeletal defect, or there are small defects across. There are small pointers which help you to know that probably it is a genetic kind of short stature. The diagnostic yield for NGS and short stature, which is described in literature, is anywhere between 10% when they did a whole exome sequencing to 42%. Of course, there is a huge variation in the kind of patients that they studied. And not surprisingly, the ones with a diagnostic yield of 42% picked up patients who are probably syndromic. They have a high number of cases with 3M syndrome, with a primordial dwarfism. So, but naturally, they started out with a cohort where we would expect a higher diagnostic rate as compared to the cohort of 10%, which only picked up children with idiopathic short stature. But there are papers which say that even when you're considering testing patients with a probable genetic short stature, rather than limiting it to a panel, and this makes sense, it's better to do a whole exome sequence. Why? Because these are again those phenotypes, like how I discussed, they are the ones where we do not know all the genes. It's not like a glycogen storage disorder where we know that, yes, these are the number of defects and this is the gene, and if I've done the testing, if it's negative, I have excluded it. But rather, these are those kind of phenotypes where we expect that more and more genes will be discovered with time. So this study picked up children who had diagnosis of growth hormone insensitivity. They first did a candidate gene sequencing or a targeted panel testing. They picked up a diagnosis in 35. The children who did not have a diagnosis went on to a whole exome sequencing, and additionally, there were 11 who were diagnosed. So it's important to understand what kind of condition you're dealing with, how well is its genetic basis understood till date. Is there a possibility that we are going to add on to the gene number with coming time? And if yes, it always makes sense to start out with a broader test. So what guides your choice of test? It is the clinical phenotype. So if the important question you need to answer is whether you're dealing with a copy number variation or a single nucleotide variation, and accordingly you branch out your test. The day is not far, and we all know that there will be a day when both copy number variations, single nucleotide variations, and methylation abnormalities will be picked up by a single test, but currently we do not have such testing technology. We can look at copy number variations through whole exome or clinical exome data, but any detected copy number variation has to be confirmed, and the absence of a copy number variation by CNV calling on an exome data does not implicate that the child or person does not have a copy number variation, because we are just taking out data and trying to examine. So if there is a certain portion which is uncovered by the data, my caller is going to call it as a deletion, but there is no true deletion. If you do think that you're dealing with a single nucleotide variation, do you need NGS? You don't need NGS for all phenotypes, and a good example for that is achondroplasia, one of the commonest skeletal dysplasias. If you just test these two mutations in the FGFR3 gene, you end up making a diagnosis in more than 99% of achondroplasia patients. So there's absolutely no need for an NGS. That's a very costly test for a patient with achondroplasia. So ultimately, you have to follow a phenotype-based approach. You could start out with a single gene. So again, if you boil down to a congenital androgen insensitivity syndrome, you would start with a single gene. A single gene could be analyzed by Sanger or NGS. But if you're dealing with a disorder of sex development, probably you would like to start out with a panel or a whole exome. Similarly, if you're dealing with something like a neonatal diabetes mellitus or MODY, again, you can do an NGS panel testing, or you could go for a whole exome sequencing. If you have an undefined short stature or an obesity syndrome, probably a whole exome would be much better. So when you're thinking about a test, you need to consider what all do you want? Well, as a clinician, I would want specific genes which have a good gene disease association to be covered in the test. I would also want differentials to be covered if it is possible. I would like to avoid any variant of significance as much as possible. Trust me, they are headaches, they are nightmares. And most importantly, I would also like that the test includes newly discovered genes so that it affords me the chance to carry out a robust reanalysis. The dictum in genetic testing is that today, if you've done a test and it's negative, it's possible that you could go back and re-examine the data and you can find a variation, or you could find a disease-causing mutation down the line. The reanalysis picks up answers in around 7 to 10% of cases. Why does this happen? Because new genes are implicated with disease and they were not looked upon the first time, because today when I did the test, that gene had no association with that particular disease. So how do you make the reanalysis robust? You require the reanalysis in around 20% of cases. Frankly, it almost amounts to that number. Now, if you start out with testing 5,000 genes, I am going to be able to reanalyze only 5,000 genes. But if I start by testing 19,000 genes, of course, when I reanalyze, I'll be studying all 19,000. So similarly, if you test only 200, I'll be able to reanalyze only 200. I can only reanalyze how much I test. I cannot look at genes which have not been included in the test platform. So the most important thing is there is a race to keep up with the gene count, which is proving to be impossible with the amount of literature that is getting published every day. Understanding all these aspects, here at Newburgh, we have come up with a test technology which is called Orion, which is an NGS-based test, which is done upon Illumina NovaSeq, the latest sequencer. And I'm proud to say that we are the only private lab in India which has the sequencer. How do we differ? Very simply put, if any sample that you send to us, we do a whole exome sequencing, which covers more than 19,000 genes. Then if you tell me that no, the phenotype is only neonatal diabetes mellitus, like I showed you, I would filter out genes of neonatal diabetes mellitus, examine them for mutations, look at them for coverage, and then release a report. If I do feel that some genes are undercovered and they are very important for the disease, depending upon how many percentage of cases are reported in literature, I would go back and backfill those areas to make sure that the genes are 100% covered. But I would say most importantly as a clinician, the test offers me a scalability. So if you start with a particular gene, like supposing I say you start with the androgen receptor gene sequencing, it's fully covered, I've got no mutations to offer you. And then you tell me, okay, you just scale it up at an additional cost to WES. It's possible because I've already sequenced those 19,000 genes and kept it in my database. So we are trying to be comprehensive. We are trying to be precise because we don't want to overload patients with a variant of unknown significance. And at the same time, being scalable makes the test cost effective. It might be very important even for well-known phenotypes. So this is a patient who has undergone an Orion analysis for adrenal insufficiency. Now, if I were to fashion a panel for adrenal disorders, I would only look upon genes which are implicated in the adrenal steroidogenesis pathway. Well, this patient had a mutation in the AIRA gene, which causes autoimmune polyglandular syndrome. Yes, I would also expect that the patient had some amount of immunodeficiency, but this was the amount of data which was provided to us. So it's possible that your patient is having a gene defect, which is very different from what is described in the common pathway. Another such gene which causes isolated adrenal insufficiency and may not be included in a panel of adrenal disorders is your ABCD1, which is responsible for X-linked adrenoleukodystrophy, where an isolated adrenal insufficiency is known, especially in young males. More importantly, and I think this is where all of us need to spend time in our clinic, that is a pre-test genetic counseling. It's important to the two main messages to give your patient is that there is no guarantee that we're going to come up with answers. A negative test does not exclude a genetic disorder, and there is a possibility that we might have to test the parents or other family members, what we term as segregation analysis, to understand the significance of variants detected. When you do any genetic test for any kind of phenotype, there is a 20% chance that I might give out a variant of unknown significance. It might be possible even if the variant is found in a gene of your interest. So supposing you are having a patient with Morquio syndrome and I find a mutation in the gene, even though you tell me that yes, it matches the phenotype, I will have to call it a variant of unknown significance because the American College of Medical Genetic guidelines are very, very strict. Unless the variant is reported in multiple patients or there are functional studies which are unequivocally proving it to be damaging, I cannot call the variant as pathogenic or likely pathogenic, though if you follow it up with an enzyme study, which again corroborates your finding of Morquio, I would call up and tell you that yes, there is a very high possibility that the variant is actually disease-causing, but the guidelines preclude me from classifying it into a pathogenic or likely pathogenic variant. So pre-test genetic counseling and post-test genetic. I think post-test genetic is very, very important, not only for the patient, but also important with the laboratory personnel to understand what else is possible within a particular sample. So when you come to variants, we've stopped calling them mutations, or rather we call them variants nowadays. We classify them according to their significance. Again, as I told you, all this variant classification is based upon guidelines given by the American College of Medical Genetics. You could have pathogenic variants, means that the variant has been so robustly associated with disease that we have no doubt whatever that it causes a problem. Or the variant has stopped a protein from being formed at such a position that I'm very sure that it will cause a disease. A likely pathogenic, I am 90% sure that that particular variant which I detected is probably disease causing. A variant of unknown significance, just as it is colored, it's very gray in nature. It's possible that it might be significant. I might be able to change its significance. And that is another thing which is very common amongst genetic laboratories. We tend to reclassify variants within time. So if you have a patient who has a variant of unknown significance and you want to consider further testing 6 months down the line, it would not be a bad idea to go back to the lab and tell them that please kindly relook and tell me if the significance has changed. Is it possible that it has become likely pathogenic, or does it still remain a variant of unknown significance? It is also important to understand that the guidelines talk about variant-specific classification. You might detect a likely pathogenic or pathogenic variant, but it is not necessary that that proves the diagnosis. So if you take up any autosomal recessive disorder like GSD type 1, it is caused when there are 2 gene copies which are affected. If I pick up only one pathogenic variant in one gene copy, that does not mean that I have diagnosed the child with GSD. It just means that there is one pathogenic variant. Either the other variant is present in a place which is not covered by the test technology, either there is a copy number variation which my test technology is missing, or it is possible that the patient has some other disorder and just by chance he has that one pathogenic variant in the GSD gene. You could also have a negative report. The best of the best figures that we give out are 40 to 50% chance of getting an answer, and an answer would include any of the 3 when a patient undergoes whole exome sequencing. So I would just end by saying that it is time to challenge the time-tested approach. So rather than adopting a sequential testing platform, probably we should do multiple tests together. I'm a big believer of biochemical analysis. I do believe that whatever initial testing you do helps us focus on particular genes, and that itself would probably increase your chances of coming to an answer. Thank you.
Thank you, Dr. Kotecha. I would request Krishna to please come.
Yes. So we have one question, doctor, in the chat box, and I would request all the participants, all the doctors to type in their questions so that we can answer all of them. So we have one question which is which cells used for karyotyping, neutrophils or lymphocytes?
Lymphocytes.
So that's the only question we had in the chat box, ma'am. So I would request
Shall I ask
everyone
one question, ma'am?
Yeah.
Yes, sure. Please go ahead.
First, I have 2 questions, ma'am. First, when we send sample, any particular precaution should be taken to prevent cell lysis and proper transport?
Usually you need to, most of the genetic samples that you send are just 4 ml blood in EDTA for molecular testing. For karyotyping, you require blood to be taken in a heparin vacutainer. Most of the times you can safely transport them in room temperatures. If in times of summer season, if it's really, really hot, then it would not be a bad idea to use a gel pack.
And what about pre-implantation genetic techniques, ma'am? Is it available in your center?
Yes, it is available. But of course, you can only go for pre-implantation genetic techniques once you have a clear-cut defined genetic etiology in the affected child. And again, just like how we say that prenatal testing is ideally not recommended in a variant of unknown significance, similarly, pre-implantation genetic testing is also not recommended, usually in a variant of unknown significance. I did share many examples where your variant of unknown significance could be responsible for a disease. So it is best to have a discussion with the lab. Again, it's important for me to tell you that not all mutations that are picked up can be tested on pre-implantation genetics. The commonest would be MLPA. MLPA requires a lot of DNA. We will not get so much DNA from an embryo. So usually whenever you discuss pre-implantation genetic diagnosis, it would also be good at the same time to send the reports over to the lab. We usually try and see what kind of mutations are there, whether they can be picked up on a pre-implantation genetic test. And we also need to do some pre-PGD workup before we directly go to test the embryo.
Sure. Thank you, doctor. We have one more question in the chat box. How many days take for karyotype?
For karyotype, it depends how fast your culture grows, but anywhere between 15 days to around 25 days. There are sometimes the culture has a very low growth rate. It could be either because there is some amount of contamination within the cells, and that might take longer, but roughly around 15 to 25 days.
Sure, ma'am. One more question we have from Dr. Rahul. Madam, could you guide on what test to request when Noonan syndrome is suspected, and what is the chance of picking up the condition?
So Noonan syndrome again has multiple genes. So you could go for a Noonan syndrome panel. Currently, I think there are around 9 genes, if I'm not wrong, which are known to be implicated in Noonan syndrome. The chances that you would pick it up, very difficult to say. I mean, I'm sorry, I'll have to actually refer for exact figures. I will refer and get back to you.
Sure. We have one more question, ma'am. How many cells do you usually see for karyotyping to rule or Turner? For mosaic, how many do you need to see?
No, for karyotyping, we look at 20 metaphase spreads. So what I showed you was one metaphase spread. So we look at 20 metaphase spreads. We usually give the picture of either 1 or 2, which are quite clear, but we count 20. So basically you're interested in the counting. That's what is important in a Turner. In fish, normally we count 50. To exclude a mosaicism, it has been suggested that you count up to 200. But of course, if I count 50 and I encounter a mosaic, then I know that I'm dealing with a mosaic Turner. So it's mainly for a negative predictive value that I count 200 cells in a fish.
Thank you, ma'am. One more question we have from Dr. Pankaj. Pre-implantation with cells and at what time gestation sample taken?
Sorry, Krishna, your voice broke.
Okay, we have a question from Dr. Pankaj. Pre-implantation with cells and at what time gestation sample taken?
I mean, there are 2 tests, right? One is your pre-implantation genetic testing is done on embryos after an IVF procedure. There, your embryologist usually picks up cells in the blastocyst stage. I think that's after around 5 to 7 days. Now, different centers have their different protocols. The other test which is commonly used, because in a pre-implantation genetic testing is a costly affair, the other commonly used genetic procedure is what we call prenatal testing, which is done either on chorionic villus biopsy or an amniotic fluid. A chorionic villus biopsy is done at 12 weeks of gestation. It's usually recommended for those disorders which have a 25% risk of recurrence, meaning that if I have a child with achondroplasia, I wouldn't be recommending a chorionic villus biopsy because the chances that it will recur is only 1%, unless his parents are affected or something like that. And for other disorders which do not have a 25% risk of recurrence, we would recommend an amniotic fluid which can be withdrawn anytime from 16 weeks till around 20 weeks. Usually the limit is 20 weeks because though the legal limits of termination have been extended to 24, you need to give the lab time to test. But I think most importantly is you cannot embark on any kind of prenatal testing till you have a definitive, and when I mean definitive, at least in the NGS terminology, it's likely pathogenic or pathogenic mutations in your patient. So you need to have a definitive etiology in an affected child before you discuss prenatal testing.
Sure, ma'am. We have one question from Dr. Tausif. What genetic testing do you do in familial dyslipidemia?
Sorry?
Dyslipidemia.
Familial dyslipidemia. Yeah. Okay. So familial dyslipidemia, of course, it consists of multiple genes. So you could either go for an we usually do an NGS. You could either do an NGS panel, or you could go for whole exome sequencing. So basically, we need to look at multiple genes together to try and come at an answer.
We have one question from Dr. Rahul. Could you also shed some light on meaning and details of in silico testing?
Oh, sorry?
In silico testing. In silico testing. Computational tests.
So you're talking about computational predictional tools, right?
Yeah.
Huh. So these computational predictional tools are basically used to try and find whether a particular genetic variation is disease causing or not. You could have multiple tools depending upon the kind of genetic variation. But what we need to understand is, and this is something which has now become very common across multiple laboratories, that rather they are only predictions. So recently I released a report of a patient of a liver disease where the tool told me that no, it is not causing a damage, but it was well reported in literature because it was a silent mutation. So the amino acid did not change, but it was well reported in literature to be disease causing. So yes, we labeled it confidently as a variant of, as a likely pathogenic mutation. What is more important, I would say, while judging a variant is how frequently is it found in normal population. We commonly look at the GenoMAD database for it. Though we do not have exact Indian population, if you look at allele frequencies in the South Asian population database, they quite well match with the Indian population. So if I have a variation in a very rare disease, and if the GenoMAD tells me that, that variation has been seen in around 50 different individuals, I will start doubting its significance because the disease is so rare that I wouldn't expect 50 normal individuals to be having the mutation, even if it is present on a single allele. So it all depends on, your disease incidence, what is your allele frequency. We use the predictions to help us, but classifications are never totally based on the prediction.
There is one more question from Dr. Santu related to this, so I'll just take it. So for VUS, does in silico analysis change the report status?
No, the in silico, so usually when you get a VUS from a laboratory, they've already done an in silico analysis. If you go through depths of the report, most of the reports will say whether the variation has been reported previously or not. What is its frequency in the normal population? What are the in silico predictions? And clubbing all that together, the laboratory makes an independent assessment. You must have also come across the term ClinVar in many of the reports. So ClinVar is a database. It is an open database for laboratories to submit variants which they have come across during testing, and they submit variants in a way that they have classified. Now, the ACMG guidelines are so strict that they say that just because laboratory A classified a variant as likely pathogenic, I am not allowed to go ahead and classify it as likely pathogenic unless the laboratory has provided enough evidence on the basis of that classification. So just on the basis of in silico tools, I wouldn't say that your variant predictions are going to change. There is a higher chance that your variant classification will change because the same variant might get reported in another patient, or it is possible that multiple normal individuals were detected carrying the variant. Each laboratory also has its own database. So today, if I saw a variant which I called as a variant of unknown significance in neonatal diabetes. And down the line, if I saw the same variant in 10 other patients with neonatal diabetes, within my laboratory, I have accumulated enough data to come back and tell you that, yes, it looks significant. Now, the biggest problem here is most of the labs, including ours, is so understaffed that we cannot reach out to you. So we rather request clinicians that you reach out to us after a particular point of time, usually at 6 months or a yearly interval, finding out whether there has been a change in variant classification.
Sure, ma'am. We have a question from Jalak. Do we need to specifically ask to see more cell if mosaic turner suspected?
You might, depending on how many cells the laboratory saw. And it is possible that the sample that you submitted, so there are some failure rates in every technology, so it's possible. So when you submit any sample, we ultimately spin it and we get a small pellet out of it, after which we do everything. So it's possible that the sample that you submitted did not lead to so many cells that we were able to count 200. So it's important to ask the lab how many cells they counted in a fish. Most laboratories will count only 50 unless you specify that you're looking for a mosaic syndrome and you would like 200 cells to be counted.
We have one question from Dr. Sela. Ma'am, as you said, it's difficult to pick up every gene in DSD, but if we get nothing in NGS, VAT to do is very difficult, especially when counseling patients. It's not VAT. So what use as a genetist advise further? Cost of MS-MLPA.
So basically, I think, see, it all depends how much pre-test counseling you have done. So every patient whom I counsel, the first thing that I say is that the chances that you will get anything in any kind of test anywhere around the world, when we're talking about whole exome, because maximum amount of data. So I would say in whole exome is 40 to 60%. In clinical exome, it's somewhere around 20 to 30% in a very well-defined phenotype. So if I know I'm dealing with a mucopolysaccharidosis and I'm very sure I've done the enzyme assay, yes, those are the cases where I do expect a mutation. And if I don't get, I'm a bit surprised. But otherwise, in broad-based phenotypes like a disorder of sex development, my pre-test counseling basically starts first with the fact that there is a high chance that you might end up not getting anything. There is only a 40% chance if the patient opts for whole exome sequencing to get a diagnosis. Within that 40%, there's a 20% chance there might be a variant of unknown significance. I also do add that yes, if we do get an answer, you're not looking out for one. I mean, it's not always treatment. Many of them just want to know what went wrong and they want to try and plan another pregnancy. But a negative does not exclude a disorder. I do tell them, and I tell the same for patients with VOUS. Many of them cannot afford any further testing. And of course, getting functional studies done in India is very, very difficult. You hardly have any labs where there are model organisms. So I tell them, let's wait for 6 months or a year, come back or share the report. We'll try and see whether we can add significance to the variant. We'll try and reanalyze the sample. At least from our laboratory, we allow one free reanalysis because we do understand that the reanalysis might be needed and might actually prove fruitful in 7 to 10%. But other than that, you can't tell much. So every patient, if you have already primed them of a possibility of a negative report, it's easier if you get one in your hand. The cost of MLPA, sorry, I think someone asked the cost of MLPA, right?
Yes, ma'am.
So the cost of MLPA would depend upon the MLPA kit. So it again depends upon how big your gene is. It's anywhere between 8,000 to 16,000 to 24,000. So different MLPAs have different costs.
MS-MLPA.
MS-MLPA. MS-MLPA also falls along the same line. I think it's around 8,000 or 12,000.
So we have one more question. CRISPR technology ?
CRISPR.
CRISPR is basically a gene editing technology. So once you have found a particular mutation, and that's plainly more in the research basis right now. Once you have found a particular mutation, the technology tries to remove the mutation from that particular place and then inserts a normal gene portion in that area. So that is what is CRISPR. So it's a gene editing technology.
We have one question from Dr. Aniket, ma'am. After careful analysis, we have detected a homogeneous chronic variant C.293 3CG in the POU1F1 gene, homogeneous or compound heterogeneous.
And this gene causes the variant has not been reported. Okay, so you are asking me what is the importance of this report. If I am not wrong, probably the variant has been labeled as a variant of unknown significance. And the only reason why it has been labeled as a variant of unknown significance, even though it has not been reported in any of the databases, is because it is not affecting an invariant splice nucleotide. So there are portions which are there in the intron-exon boundaries. And just at the boundary, we have what we call a splice site from where the intron is removed. And that's how the protein is formed. Now, there are nucleotides at the splice site, but the most important ones are the ones which are present on -1 and -2 region and +1 and +2 region. So when there is a mutation in a -1 or -2, or a +1 or +2 site, there is a higher chance that it is actually affecting gene splicing, and hence it might cause disease. But your current mutation, which you have sent across, is present in -3 site. So the laboratory, it is difficult for the laboratory to prove that it is actually affecting splicing, and probably that is why it has been called as a variant of unknown significance. You have two options, both of them which are very difficult, considering that your patient, I think, has some hypopituitarism, right? POUF1 is your gene, yeah.
Yes,
so
yes.
One is, I mean, if you really want to prove a splicing defect, you actually need tissue from the affected portion to prove that it is causing a gene problem, which of course I know you're not going to give in such a patient. It's impossible to attain. The other would be, the option would be to wait and watch till a patient with a similar mutation actually is reported. The third would be to contact a research group, which is actually working on hypopituitarism. So you have to understand that with the advent of NGS, probably one thing that we have lost is a publication of single case reports, which could add to a lot of data. So there might be a patient who is there in some laboratory somewhere, but the only reason that we don't know about him or her is because the data has never been published. Not all laboratories submit to ClinVar. It's a very painstaking process. So you could contact a research group and ask them that whether they have come across such a patient. And then, of course, you could take it further depending on what they suggest. There are many research groups which also say that you could send skin fibroblasts or blood, and they would try to test for gene expression there. So if it's whichever way we are able to prove that the gene expression has been affected, it would be easy. In the Indian continent, I think you have only two options: wait and watch or contact the research group.
Okay, ma'am. Thank you.
Okay, next question we have, ma'am. What's the cost of whole genome sequencing?
The whole genome sequencing, I think, in my laboratory costs around 90,000. It would cost anywhere from 90,000 to 1.5 to 2 lakhs.
Can you elaborate molecular testing in thyroid cancer?
Sorry? Thyroid cancers. For thyroid cancers, you're talking about multiple endocrine neoplasia? So if you want to analyze genes together, you can go via the NGS testing. Now, when we talk about NGS testing, so when you talk about cancer, there are two kinds of mutations that you're trying to see. One is you're seeing whether there is a presence of a somatic mutation, that is a change in any of the genes within the thyroid tissue, That would require testing on thyroid tissue. And the other would be that you are actually looking at genes in a patient's blood to see whether he has any condition which could lead to a thyroid carcinoma or germline mutation. So if you want to test a somatic mutation, the testing is entirely different. We have different panels for that, somatic cancer panels for specific mutations in thyroid tissue. And if you want to test for germline mutations, you could do it on blood. Sorry, what are the other questions? We can enable the joint box. I think then the other question is, ma'am, is a case of Modi panel, do you do all gene analysis in one go or you do depending on probability? Well, Dr. Tausif, in my lab, as I said, we just follow a simple blanket rule for all cases, whether it is Modi or whether it is neonatal diabetes mellitus. I'm going to sequence at least all the 19,000 genes as a part of a whole exome sequencing. I will analyze only those genes first which have been reported in literature with Modi. If I do not get anything, then I start looking at all regions which I have sequenced to see if I hit upon something which probably has been implicated either in a genetic Modi pathway or has been seen in any animal model or something, but that then such variations would actually be called as a variant of unknown significance. What you will prefer to pick Y chromosome in Turner, FISH, or SRY gene on NGS? You don't need to do for Turner, a FISH for Y chromosome would be more than enough. If you do it on blood, that again is more than enough in most cases. However, if virilization is present, in a case of Turner syndrome, then it is necessary to do the same FISH test, repeat the FISH test, which you've done on the blood sample, also on a buccal swab and on skin fibroblasts. I wouldn't go to test SRY gene on NGS. Skin fibroblast culture indications and advantages over other tests in DST. Commonly, we don't do skin fibroblast culture. Skin fibroblast culture is an invasive procedure. The only reason why you would do or consider a skin fibroblast culture is when you're dealing, again, as I said, with a mosaic condition, where I want to try and look at other tissues to see whether the patient has a tissue-dependent mosaicism. So there is mosaicism, but the number of cells in blood are so few that even when I counted 200, I couldn't pick up the mosaicism. But you, as a clinician, are 100% sure that it is Turner and it is nothing else. Then I will reach out to you and tell me that please send me a buccal swab or please send me a skin fibroblast. I will culture it and then I will see whether there is a possibility of the Turner syndrome in that skin fibroblast. The other reasons why I would be doing a skin fibroblast culture could be to study gene expression patterns. So skin fibroblasts also serve to be very good mode to functionally study gene expression, depending on whether the particular gene is expressed on the skin fibroblast. So we can't do it for each and every gene. The gene has to be expressed in skin fibroblasts normally. We have to know what is the rate of expression. Only then when I study it in a particular patient, I can tell you whether the expression is reduced or not. So I wouldn't say that it has any specific advantage as compared to other tests in DST. Basically, it's an invasive procedure. It's not easily accepted by patients. There is a very high chance of bacterial contamination when you transport skin fibroblasts, and culturing them requires a lot of effort. So the culture is costlier than a normal blood culture. And more importantly, there are many cases of skin fibroblast culture failure because of low level of contamination, because skin itself is crowded with commensal organisms. For hypopituitarism, would you suggest a target gene panel or clinical exome? I'm not a big fan of clinical exome. I would rather say that if I start out with a target gene panel, and if it's negative, I would prefer to go to whole exome. As I showed you, there is not much difference between a clinical exome and a whole exome. If you are interested in studying 10 genes, Why should I opt for a test which is going to sequence 5,000 genes and give me 10 genes? Why not I opt for a test which sequences 19,000 genes and then studies those 10 genes? At least if it is negative, I can elevate the test and study all 19,000. If you have done a clinical exome, you start out with a targeted panel. See, a lot of it also depends upon how well affording the patient is, whether the patient has any markers which help me say that it is, particular gene. That is what also decides testing. But supposing you start out with a targeted panel, it is negative, a clinical exome has been done, you scale it up to clinical exome, again it's negative, you will have to again explain to the patient that you will have to do a whole exome. And at least in India, explaining multiple tests again and again is a very big headache to patients. Thank you. I have given you my contact email ID as well as number in the slides. We will be circulating articles which I had reviewed before this presentation. If anyone is interested in a particular topic and you want in-depth information on that, we will try and search out reviews and get back to you with articles. We are also in the process of preparing an information leaflet where we plan to list out the various endocrinological disorders and the tests available and how you should be approaching. But let me tell you, there's no blanket approach. It all depends how the patient presents. And of course, when you have to choose amongst the various NGSs, as I said, it would all depend how much genetic information is known till date. If you're dealing with a phenotype where you expect new information in the coming days, it's always better to start out with a broader test. Thank you.
Thank you so much, ma'am. Thank you everyone for joining us today. In case of any questions you may have, you can always reach out to us, to ma'am's email ID and the mobile number. And
where is email ID and mobile number, please? It's not there in chat.
Can you display it once?
Okay, just a minute. Or should just a minute, it's the last slide. Sorry.
Email ID is udaya.kotecha@supratechlabs.com, udaya.kotecha@supra.
Put it in the chat box.
I'll put it in the chat box only. Okay.
I think yes.
You put it in the chat box. Okay.
Yes, I did that, ma'am.
Oh, okay. Sure, we'll send those articles and we'll send the leaflet. The leaflet will take some time to prepare, give me around 15 days. And we'll send over the articles by today or tomorrow. Thank you so much.
Thank you, everyone.